So, my name is Alice Toderi. I will host the webinar today. And before to start, I would like to give you a couple of information. So the webinar is being recorded and will be available on the website of biostimulant.com and on our social media in the next days. I invite you to use the Q&A box to submit the questions during the webinar, and we will be collect them and answer at the end of the presentation of our guest. The topic of today's webinar is biostimulants and plant nutrition. And it is my pleasure to introduce today's speaker, and you will excuse my pronunciation, Dr. Sang-Han Choi. Sorry, but he will tell us better. So Dr. Choi is a postdoctoral research associate in Dr. Leskovar's lab in Texas A&M AgriLife Research Center in Uvalde, in Texas. Dr. Choi received his PhD from the Department of Horticulture at Purdue University in 2021. He specializes in sustainable horticultural crop production in controlled environment agriculture and in the field, focusing on abiotic stress, antioxidants, resource use efficiency, nutrition, and biostimulants. So welcome, Dr. Choi. The floor is yours, and you can start whenever you feel ready.
Hello, can you hear me?
Yes, perfect.
Okay. Can you see the screen?
I think screen is perfect.
Okay. Hello, everybody. I'm Doctor Choi. I'm a postdoc research associate in Dr. Daniel Leskovar's lab at Texas A&M AgriLife Research. First of all, thank you for providing me this opportunity to give seminar. It's my honor. Today's topic is biostimulant and plant nutrition. Can you hear me clearly?
All right. I can hear you, so I think that everybody's fine.
All right, let me start with background first. As the world population continues to grow, so does the demand for fresh, nutritious food. But we are facing major challenge. There is only so much arable land available. That's pushing us toward more intensive crop production. One promising solution is controlled environment agriculture, or CEA. With its ability to produce high yields in a small area and its potential for precise crop management, CEA is becoming more and more important. But there is a catch. To ensure high productivity, growers often apply large amount of fertilizer. Unfortunately, that can lead to problems like leaching and runoff, which contaminate nearby water bodies. At the same time, there is a growing global momentum around sustainability, and greenhouse production isn't exempt. So we are being called to find ways to produce crops more responsibly. So the question is, how do we meet growing food demand while protecting our environment? That's the question that has driven my research. My work focuses on reducing excessive fertilizer use without compromising yield, quality, and nutrition. Let's talk about nitrogen fertilizer a little bit. Each year, around 85 to 90 million metric tons of nitrogen fertilizers are applied worldwide. But here's the problem. Roughly 50 to 70% of the fertilizer never been reached to the crops. It's lost through runoff and leaching. That excess nitrogen ended up in nearby water bodies where it caused serious environmental issues like nutrient pollution and eutrophication. On top of that, the cost of nitrogen fertilizer has been rising, putting even more financial pressure on growers. So now more than ever, farmers are being challenged to find smarter ways to manage fertilizer. ways they reduce waste and environment impact while still protecting their bottom line. So how can you reduce nitrogen fertilizer use while still maintaining healthy plant growth? Well, several strategies have been explored. We have precision irrigation, which help deliver water and nutrient more efficiently. There are also crop improvement through genetic modification, which can enhance nutrient use efficiency. But recently, a new approach has been gaining attention: biostimulant. What's really exciting is that biostimulants have shown benefits under both low and high nitrogen levels, making them a flexible tool for sustainable farming. Because of their potential, the biostimulant market is booming. It's projected to reach $7.6 billion by 2029. So it's clear biostimulants aren't just a trend, and they're a promising piece of future of sustainable agriculture. There are many different types of biostimulant available on the market today, but broadly speaking, they can be grouped into several main categories. Things like humic substance, microbial inoculant, inorganic compounds such as silicon, seaweed extract, chirosan, and other biopolymers. In my research, I have focused especially on one group: protein, hydrolysates, and amino acid. Protein hydrolysates, I'm going to say pH. pHs are a mixture of polypeptides and free amino acid. They are produced by breaking down protein sources through partial hydrolysis. Now, these protein sources usually come from either plant or animal origin. And the interesting part is that they're mostly agricultural byproducts. So, in a way, we are reusing agricultural waste to make something useful. Animal-based pH typically require high energy input, things like high heat and pressure. On the other hand, plant-derived pHs are processed using proteolytic enzymes, which is a much lower energy process. In my research, I've been working with a plant-derived pH product from Hello Nature company, which is derived from legume seeds. Plant-derived protein hydrolysates have been reported to show hormone-like activities that promote plant growth, boost yield, and enhance nutrient uptake. Because of these benefits, pHs have strong potential to help us address fertilizer overuse, especially in controlled environment agriculture. But there are still a couple of gaps. First, we know that pHs have hormonal activity, but the specific mechanism behind those effects haven't been fully explored yet. There's still a lot to uncover. Second, in the greenhouse production, growers typically use commercial growing media, not field soil. But most of the pH research so far has been done using field soil or media that aren't commonly used in commercial production. That makes it difficult to translate findings directly to real-world greenhouse production. And lastly, hydroponics are becoming more and more popular in greenhouses, especially for leafy greens like lettuce. But there is almost no research on how pH affect hydroponic lettuce, particularly under different nitrogen levels or nitrate to ammonium ratio. So with all that background in mind, I designed 3 studies to dive deeper into these gaps. Let's start with the first study, which focused on understanding the hormone-like activity of protein hydrolysates in promoting adventitious root formation in cutting. The findings of this study was published in Agronomy Journal. Let's talk about a little bit of background. pHs are known to have hormone-like effects that support plant growth and yield. In fact, studies have shown that pH demonstrate auxin and gibberellin-like activities, especially in crops like tomato cuttings and pea plants. So these effects are thought to come from bioactive peptides present in the pH formulation. To dive deeper into the hormonal activity of pH, we tested how plant-derived protein hydrolysates affect morphological and metabolic changes during adventitious root formation. We used stem cuttings from 3 herbaceous species and prepared treatment solution at 4 different pH concentration and 4 oxygen concentration along with a control. The cuttings were dipped into the treatment solution for 3 seconds and then placed into the inert rooting medium. They were grown in the mist room for 20 days, and at harvest, we evaluate both morphological traits and metabolic markers to evaluate the rooting response. Here's the result. These graphs show the dose response curves for shoot dry mass, root dry mass, and total root length. The empty circles represent basil, the black circles are tomato, and the triangles are chrysanthemum. On the left side, we see the response to the pH. Across all 3 species, the pH treatment resulted in a steady dose-dependent increase in shoot and root growth, as well as root length. In contrast, oxygen-treated cuttings show on the typical biphasic response, improvement at low concentration but negative effects at higher doses. Also, it's worth noting that pH require higher threshold concentration to trigger rooting responses compared to the oxygen. So while both pH and oxygen influence growth, their effects and how plants responded to them are clearly different. This table shows the relative root diameter class length. We divide the roots into the 5 categories based on the thickness using 0.25 milliliter intervals. So the category 0 to 0.25 millimeter groups represent the finest roots, while anything above 1 millimeter falls into the thick root category. On the left, you see the 5 treatment control to concentration of pH and 2 of oxygen. Although there are plant-specific responses, what stands out here is that pH consistently increase or at least tend to increase the length of the fine roots across all 3 plant species. Oxygen, on the other hand, didn't show same pattern. So this suggests that pH and oxygen influence root architecture in distinct ways. To understand metabolic responses, we measure DPPH and ABTS antioxidant capacities, along with total phenolic compounds in both roots and shoots. These are good indicators of antioxidant metabolism. Although there are quite bit variation overall, we did observe some clear trends. In particular, in basil shoot and chrysanthemum root, pH and oxygen showed opposite effects. pH treatment increased antioxidant activity and phenolic content, while oxygen actually suppressed them. So this contrast further support that pH and oxygen act through different metabolic pathways. We also look at another metabolic response, the endogenous levels of steroids in both shoots and roots. Stigmasterol, beta-sitosterol, and camptosterol are key precursors in steroid biosynthesis. In our study, non-treated cutting showed the native steroid pool, and we found relatively high steroid level in basil, followed by tomato, then chrysanthemum. Interestingly, both pH and oxygen show either no change or similar directional changes, meaning they either increase or decrease steroid levels in plant tissues. These changes were relatively minor in basil, which naturally high steroid levels, but more pronounced in chrysanthemum, where the baseline steroid levels were lower. So this suggests that pH and auxin might share some overlap in how they influence steroid biosynthesis. With this result, here's the conclusion. To conclude the first study, we investigated the hormone-like activities of plant-derived protein hydrolysate in adventitious root formation of cuttings. So contrasting effects of pH and auxin in dose response curve of growth, root morphology, and antioxidant activities were observed. Native steroid pools of plant species influence adventitious rooting response to the pH and auxin. The similar effects of pH and auxin on endogenous steroid content demonstrate that their overlapping role in steroid biosynthesis. pH-induced, so in conclusion, pH-induced adventitious rooting responses of cutting via steroid mediated process while interacting with auxin. So the second study was to examine the effects of pH application methods on greenhouse lettuce and tomato yield and quality under different nitrogen level. The outcomes were published in the Agronomy Journal. Let's talk about the background. Previous studies have explored the effects of plant-derived protein hydrolysates under different nitrogen levels in crops like spinach, lettuce, rocket, and tomato, and typically grown in the field soil or quartz sands. But in greenhouse condition, growers commonly use commercial growing media, not field soil. On top of that, pH can be applied either through foliar spray or directly to the roots, but we still don't have clear information on which method works best in greenhouse setting. To fill the knowledge gap, dwarf tomato microtome and romaine lettuce dragon were grown in greenhouse condition. Plants were grown in 6-inch standard pots filled with commercial growing media and fertigated with drip irrigation using a 0.625 strength Hoagland solution with 4 different nitrogen levels, from 2 millimolar to 15 millimolar. The pH treatment were untreated control, foliar spray, and root application. They were applied every week. Lettuce and tomato plants were grown for 4 and 8 weeks after transplanting, respectively, before they were harvested. This table shows how different nitrogen level and pH application methods affected the growth and lettuce and tomato. The red shaded area at the top represent tomato data, and the green is for the lettuce. Overall, both nitrogen level pH application significantly influence plant growth. Especially, nitrogen level was a major driver of growth responses. Increasing nitrogen level consistently improved shoot dry weight, fresh weight, and leaf area in both crops. Now, what's interesting is the effect of application method. The root application of pH increased tomato fruit dry weight by 22% and lettuce shoot dry weight by 31% compared to the untreated control. The foliar spray the foliar spray PHF had a positive effect too, but it was somewhere in between. So under greenhouse condition using commercial growing media, root application of pH consistently outperformed foliar spray of pH in boosting plant growth. These figures show chlorophyll and carotenoid concentration in the leaves of lettuce and tomato. In these graphs, the black circle represents the control, diamond indicates foliar application, and the triangle shows the root application. Both nitrogen level and pH application methods have significant effects on chlorophyll A, B, and carotenoid content. When we averaged the data across all nitrogen level, root application of pH consistently resulted in higher pigment concentration than foliar application, regardless of nitrogen level. So this suggests that root application of pH may enhance photosynthetic pigment production more effectively, which could contribute to improved photosynthesis and overall plant performance. So we wanted to know, does it really increase photosynthesis rate? Here is the result. Photosynthesis rate and water use efficiency were measured in lettuce and tomato 20 days after transplanting. In these graphs, the top row shows photosynthesis rate and the bottom row shows water use efficiency. Root application of pH significantly improved both photosynthesis rate and water use efficiency in lettuce and tomato compared to the untreated control. And just like with pigment concentration, root application was consistently more effective than foliar application. So this result suggests that root application of pH not only improved photosynthetic activity, but also improved how efficiently plants use water. This table shows the results for antioxidant activity and compound. What stands out here is the root application pH significantly boosted antioxidant capacity and compound in both lettuce leaf and tomato fruit. Especially in tomato, it also increased lycopene levels. The foliar spray did help too, but effect was somewhere in the middle. So overall, root application turned out to be more effective than foliar spray in increasing the nutritional quality of the crops. When you look at the nitrogen level, higher nitrogen level reduced antioxidant activity in the lettuce, but not in the tomato. This really highlights how over-fertilizing with nitrogen can negatively affect food quality, especially in leafy greens. It was tested whether pH increased nitrogen use uptake to reduce fertilizer use. The equations are presented on the right. We found that root application of pH increased nitrogen use efficiency and uptake efficiency in both tomato and lettuce, regardless of nitrogen level. So each effective across different crops. Interestingly, the biggest improvement in the nitrogen uptake happened at low nitrogen levels, which show that root application of pH is especially helpful when nitrogen is really limited. That's a big deal for the sustainable production, maintaining yield while cutting back on fertilizer. And here is the practical takeaway. Going from 10 to 50 millimolar nitrogen didn't improve yield or efficiencies. So with root application of pH, you can use moderate amount of nitrogen and save on fertilizer costs and reduce leaching risk while growing high quality crops in the greenhouse settings. In the second study, the effects of pH application methods on yield and quality of greenhouse lettuce and under different nitrogen levels were examined. The root application of pH outperformed foliar spray by increasing yield, photosynthesis, water use efficiency, chlorophyll content, and antioxidant compounds and activities, regardless of nitrogen level in lettuce and tomato. Such improvements were associated with increased end use and uptake efficiencies. So root application of pH provides a sustainable solution for greenhouse crop production, helping produce high-quality crops in commercial growing media under both high and low nitrogen conditions. The third study is to evaluate the effect of pH application methods on hydroponically grown lettuce growth and yield under different nitrogen level and nitrate to ammonium ratios. It was published in the Journal of the Science of Food and Agriculture. So let me talk about the background a little bit. The plant Plants mainly uptake nitrogen in the form of nitrate and ammonium. And getting the right balance of nitrate to ammonium ratio is really important for both crop growth and quality. Now, previous studies on protein hydrolysates and nitrogen mostly used a fixed nitrate to ammonium ratio. So while we know pH can be beneficial on the different nitrogen level, um, we still don't know how it performs when the nitrate ammonium ratios changes. So especially in hydroponics, where nutrient delivery is highly controlled, understanding how pH works under different nitrogen level and nitrate ammonium ratio is something we haven't explored yet. To fill this knowledge gap, I set up an experiment using romaine lettuce grown in deep water culture hydroponic system. The plants were grown with 0.6 to 5 strength coagulant solution and tested with two different nitrogen levels, each combined with 4 nitrate to ammonium ratio. They were provided factorial design. For the treatment, I compared untreated control with two types of protein hydrolysate application, foliar spray and root application. The hydroponic lettuce were grown for 4 weeks after transplant and they were harvested. This figure shows a two-way interaction between the nitrate to ammonium ratio and pH application on shoot growth and chlorophylls. As the nitrate to ammonium ratio decreased, especially 50 to 50, 25 to 75, we saw clear drop in shoot fresh weight, dry weight. But when pH was applied to the root, it helped recover that loss, bringing shoot biomass back to levels similar to the more favorable ratios like 75 to 25 or 100 to 0. So root application of pH really helped buffer the stress caused by lower nitrate availability. As you can see here, these three factors together had a significant impact on root traits like root dry weight, total root length, root to shoot ratio, and average root diameter. One clear trend we noticed was that as nitrate to ammonium ratio decreased, that is, we added more ammonium, the root dry weight and total root length also went down. The root dry weight and total, so you can see the decrease there. The plant didn't develop as much root mass under more ammonium heavy conditions. Interestingly, the average root diameter only increased when we apply pH to the root, especially under 10 millimolar nitrogen conditions, and at nitrate ammonium ratio of 50 to 50 and 25 to 75. This suggests that the combine of pH applied to the root and ammonium-rich nitrogen sources lead to the thicker root. So overall, the root architecture wasn't just affected by one factor. It was shaped by how all three factors interact with each other. It is a well-known fact that high nitrogen levels tend to reduce antioxidant compound in plants. In our study, a lettuce grown with 2 millimolar nitrogen had 1.3 times higher total phenolic compounds and 2.1 times higher flavonoid compounds compared to those grown with 10 millimolar nitrogen levels. What's interesting is that pH application improved both TPC and TFC regardless of nitrogen level or nitrate to ammonium ratio. So even under high nitrogen, pH still helped boost antioxidant quality. On the other hand, nitrate accumulation in leaves increased significantly as we increased nitrogen level and nitrate to ammonium ratio, but pH did not statistically affect nitrate levels, and there were no interaction effects between pH and other factors for nitrate concentration. The mineral content in both shoots and roots was strongly influenced by three factors interaction. In the shoot, the nitrate ammonium ratio alone had a huge impact on overall mineral composition. The 100 to 0 And 75 to 25 ratio generally gave us the high nutrient levels, except for boron, which actually increased under more ammonia-rich conditions. We saw that applying pH, especially under low nitrogen levels, significantly helped increase uptake of nitrogen, potassium, and iron. This is showing three-way interaction of the root minerals. Nitrogen concentration in shoot minerals shows strong change in nitrogen level, but nitrogen concentration in root stayed fairly constant. So this suggests that nutrient allocation is regulated differently. Roots responded more to the pH and nitrate ammonia ratio than to the nitrogen level itself. pH improved root nutrient uptake under especially root application improved root nutrient uptake under both low and high nitrogen conditions, but in different ways. Under low nitrogen, low nitrate ammonia ratio, root application pH increased the uptake of several key minerals such as phosphorus, potassium, magnesium, calcium, and copper. When nitrogen levels were higher and nitrate was more dominant, root application still enhanced uptake, particularly phosphorus, magnesium, and calcium. So this showed that root applications of pH help roots stay efficient as absorbing nutrient across a range of nutrient conditions, whether nitrogen is limited or more available. In study 3, we examined the effects of different pH application methods on hydroponic lettuce with different nitrogen level and nitrate ammonia ratio. A root application pH was more effective than foliar spray to enhance yield and quality compared to the untreated control. Root application prevent yield loss by low nitrate ammonia ratio, and they also increase chlorophyll and antioxidants under high nitrogen level. When high nitrogen level is provided, plants usually show low antioxidant level, but root application increase antioxidant levels, even though under high nitrogen level. The effectiveness of pH is strongly affected by nitrogen level and nitrate to ammonium ratio, so the interaction is really important. Root application of pH improved nutrient uptake through the strong three-way interactions without increasing leaf nitrate levels. In this series of studies, we explore the use of plant-derived protein hydrolysates as a sustainable tool to enhance growth, quality, and nutrient efficiency in greenhouse crop production. Study 1 showed that pH influences hormone pathway in plants, particularly modifying steroid related process while interacting with oxygen during root development. Study 2 showed that applying pH to the root zone, root application can be an effective strategy for boosting crop yield and quality in commercial greenhouse media, regardless of nitrogen levels. Study 3 showed that Study 3 extended this to the hydroponic system where root application significantly enhanced root growth, which in turn supported better shoot performance and antioxidant quality. These effects were strongly influenced by nitrogen level and the nitrate to ammonia ratio. In conclusion, these findings highlight the hormone-like activity of pH and its potential as a sustainable input. By optimizing pH application and nitrogen management, we can support high-quality, resource-efficient production in controlled environment agriculture. First, I want to sincerely thank my previous advisor, Dr. Haiji Kim, for her incredible support, guidance, and patience throughout my PhD journey. A special thank you goes to my long-time collaborator, Dr. Giuseppe Colla, Dr. Maria Teresa Cardarelli. I've been fortunate to work closely with them, and our collaboration has not only continued but grown over the years. I learned a great deal from their experience and insights. Thank you very much. I also thanked Dr. Kang Mo-koo. It was a privilege to collaborate with him, and I'm truly grateful for the lesson he taught me about how to think critically and approach research with purpose. Big thanks to the previous lab members, Tang, Iru, Yuting, Peng, Gaotian, and Hesu, also Purdue greenhouse staff for their support during my experiment. Lastly, thank you to the horticulture department at Purdue University, providing me such a supportive environment, and Hello Nature for the supply, providing the protein hydrolysates for my research. Thank you all. Before we move into the Q&A, I'd like to briefly share about my current lab. I'm currently a postdoc researcher at Texas A&M Agri Research, working in Dr. Daniel Lescobar's lab. Dr. Daniel Lescobar is the center director and also the current president of ASHS. He has a long-standing background in biostimulant research and has collaborated with many leading companies in the field. So if you are interested in collaborating, please feel free to reach out to Dr. Daniel Lescobar or me. Our lab is always open to future partnership and collaborative opportunity. Thank you all for your attention. I'm happy to open the floor to any question. Thank you very much.
Thank you very much for this rich and insightful presentation. I think it was very interesting to see the different type of application of the vegetal protein hydrolysates and also the effects, not only on plants, but also on the nutrient content, which could also affect human health. So I think that your research is really key for the future of biostimulants. So I now open the floor to questions. We had a couple of questions about the slides, if the slides were available, if the recording would be available. So I'll tell here. So the recording of the webinar will be available soon on the biostimulant.com website and on the YouTube channel, etc., etc. So if you follow us on social media, you'll see the post when the recording is available. We will not share directly the slides because, of course, these are thanks to Dr. Choi. So we will not just share the slide. We'll just share the recording. So I have a couple of questions here. So one. So one is, what about the seed treatment of protein hydrolysates? So I think that the question is, have you ever tried to apply protein hydrolysates as a seed treatment, and which were the results? So do you have any experience on that?
No, I don't have experience of seed treatment. I know biostimulant can be applied seed treatment, foliar spray, root drench in many different ways. And then, yeah, so I only tested foliar spray and root drench. And then if I have a chance to test seed treatment, I would like to do it.
Okay. So I ask everybody to put the question, if you have a question, just put it in or in the chat or in the Q&A box. So we will not allow you to speak. So please write your question. So we have a question which is quite controversial. So I will ask it to you anyway, but feel free not to answer because I think that the answer is not so easy. So do you think biostimulants can be used against biotic stress, mostly against fungi, for example? So
Come again, please.
Do you think biostimulants can be used against biotic stress? So it's a plant protection product, in fact,
yes,
which is out of the definition of biostimulants product. So
yes.
I would say no for definition.
Yes, that's a really good question. So I usually test biostimulant abiotic stress conditions, but I saw some papers showing biostimulant can be effective under biotic stress conditions. one of the good example is the silicon. So I saw also another seminar, a webinar provide plan biostimulant.com. It was about the silicon, and then silicon usually help against uh, biotic stress. So I think yeah, biostimulant can be used for biotic stress, but depending on the biostimulant product you're using, it will show different effects.
Okay, thank you very much. Indeed, it's a complex question, so we would need a webinar just for that probably. So one other question from is just out of curiosity, we talk a lot about greenhouse experiments on protein hydrolysates.
Mhm.
Do you think their use is sustainable also in open field?
So the question was, if the biostimulant, especially the protein hydrolysate, will be effective in the field condition, right?
Exactly, yes.
Yes. Actually, I didn't test it in the field condition, but the reason I studied this experiment, the studies, um, is that because a lot of papers, especially on the protein hydrolysate, they show the beneficial effects in the field conditions. So I was like, what about greenhouse condition? That's how I started these studies. So I'm pretty sure especially protein hydrolysate, uh, is really effective under field condition.
Perfect.
Mhm.
Another question, how long does the effect lasts? What should be the frequency of application in case of root application or foliar application?
Mm, I think for my research, I applied every week. Um, but depending on the crops, it was recommended the different frequency, and then frequency was recommended. So I think depending on your crop, you gotta decide what frequency you're gonna apply. So for my research, I use every week application.
Okay. Yeah, for sure. It depends a lot on the crop you have. Um, so I have one question which I don't really understand is, can you tell me if I want to do PhD in formulation of biostimulants or something related to biostimulants? Um, I imagine you want some suggestion on which study to bake to become formulators of biostimulants. You have any advice for people that want to specialize in this field?
I don't get the question. Sorry about it.
It's about I think it's about education.
Uh-huh.
So if you recommend some education pathways to become um, formulator of biostimulants or study biostimulants.
Hmm.
So if you want, maybe you can share your experience, Bob.
Yes. So so when I first dive into the biostimulant, so so yeah, okay. So I was able to talk with a professor when biostimulant was introduced in United States very long time ago, very long time ago. They think that this just it's not working. Maybe it's like snake oil. So
Yes.
yeah, but um actually, when I tested and then I saw beneficial effects there. And then now I was able to talk with the professor, and then he said that, yeah, he said I also study biostimulant too. So, so he also saw but the experiment he got was like 20 years ago. So, but everybody know biostimulant is very effective. And then there are many different types of biostimulant available in the market. So um, I have tested several like humic substance, protein hydrolysis, silicons, and then microbial inoculants. And then I saw some beneficial effects. And then, yeah, I'd like to study more about it and then provide education about biostimulant if possible.
Okay, well, I think that it's all. I don't see any other question. So it's already been 45 minutes. I think that yeah, so one last question is, why in your experiment you use only protein hydrolysates?
Yes, so why I only use the protein hydrolysate? That's the biostimulant I was working on, focusing on. But so only these three study I only use the protein hydrolysate. But this study was done when I was doing my PhD degree. But now I'm in the Texas A&M Agri Life research, and then I'm testing humic substance and silicon, and then other like beneficial fungi and bacteria. Yes.
Well, we'll look forward to see your next works, maybe here at biostimulant.com. So thank you very much for this session. It was very, very, very interesting. I want to thank everybody for attending the webinar today. If you still have questions, please submit them directly on the website biostimulant.com, because there is a specific section, Ask the Experts, and we will get back to you. At the end, I would like to remind you that all the recording will be available soon. So Dr. Choi, have a nice day. Thank you very much for being here today with us. Hope to see you again, you and also our attendees here at the webinar by biostimulant.com. Have a nice day, everybody. Thank you very much.
Bye. Thank you. Thank you for the opportunity. Everybody have a good day.
Thank you. Bye.